That's all good to hear. One new type of these vaccine platforms that you mentioned is mRNA or messenger RNA vaccine, and I personally find this advance potentially very exciting because if this new type of vaccine works as well as it seems so far, then it could be possible to build on top of this advance to develop other kinds of vaccines for future diseases a lot more quickly and effectively. But I'm curious if you can walk us through how these types of vaccines work and why this new type of approach can be such a positive development, not just in this case, but more broadly, potentially.
Well, so the way it works is that it eliminates so much of the things that had formerly been tried and true. So the standard classic way of making a vaccine is you have to isolate the pathogen in question, whatever that might be, a new virus or what have you. Then you have to grow it up efficiently in either eggs or cells. That takes a long time. It's fraught with things going wrong. You grow it up, then you purify it, and you either attenuate it to give a live attenuated vaccine or you inactivate it to give sort of a killed vaccine. That's the standard way of doing things. Over the years, given molecular biological approaches and the advances in technologies, we've gotten to be much more sophisticated, not requiring having to grow up the virus. With messenger RNA, RNA is the molecular means of coding for proteins. So if I want to vaccinate you, Mark, with a component of the COVID-19 virus, the SARS-CoV-2, the important component is that spike protein. Because when you look at the virus under electron microscope, you have these spikes that stick out. That's the reason why you get the word coronavirus standing for crown. It looks like the spikes on a crown. Well, that particular spike protein is the thing you want the body to make an immune response against. So rather than getting involved in handling the virus, what happens as soon as the virus is discovered, the technology today allows us to get the full sequence of the virus in days as opposed to years. So then you publish the sequence, and anybody in any lab can pull the sequence out of the gene that specifically codes for the spike protein. So you don't have to touch the virus. You go in silico, in a computer, you pull out the gene and you stick it into this messenger RNA. Now, the purpose of messenger RNA in your body is to code for all the different proteins that your body makes. So now you're going to take an artificially made RNA, you're going to stick this particular sequence in, and then you inject it into the body. It goes into a cell, and it starts pumping out protein. So the body sees the protein and says, I think that's the virus because it's a spike protein. But it isn't the virus. It's a harmless protein that you made very quickly. So to give the viewers and the listeners an example, on January the 9th, the Chinese put on a public database the sequence. On the 10th, we had a meeting with my team that said, we're going to take that sequence, we're going to partner with the company Moderna, and we're going to maybe 3 or 4 days later meet and get the vaccine starting to be made. It's very simple. I mean, it isn't growing a virus. You just stick it in and bingo, you start making and producing the messenger RNA. Well, about 63 days later, 62 days later, we had enough of it to do a phase 1 trial, an absolutely world indoor record to go from the time you get a sequence to the time you have enough vaccine to do a phase 1 trial. A few months later, we were in a phase 3 trial. And then because there was so much infection in the community, a couple of months later, we started the phase 3 trial on July 27th. By the time you got to August, September, October, we had enough data so that by the time we got to November, we said, oh my goodness, we have a highly efficacious safe vaccine that we're now going to start to administer in December. The technology that allowed that, Mark, is beyond unprecedented. It's something that if you had told me that 10 years ago when we were making vaccines for different things, I would have told you, Mark, I respect you, but there's no chance in the world that we're going to be able to do that. And it was done. And your point is an excellent point. If we're successful, which we're there, we just have to go through the motions of proving it now with large numbers of people, that we're able to really crush a disease like COVID-19, the way we did with smallpox and polio and measles, with this new technology, you're absolutely correct. It's such an adaptable technology that it would likely transform the way we look at and make vaccines.
So I've heard from a lot of people who have questions about a new type of vaccine that involves RNA or DNA, and why we can expect that this can be safe without having any issues down the line. So I'm curious to hear how you'd explain that scientifically and your thoughts on that concern overall.
Yeah, because we know what happens, from a lot of work in the animal models. So that RNA that pumps it out for a very limited period of time, enough to get a good immune response. And then the RNA degrades the way RNA generally degrades under most circumstances. So what the FDA has done is that they are not going to consider an EUA until 60 days following the time that half the people in the trial have received their last dose. And the reason the 60 days is the magic number, because when you look at the experience over decades with vaccines, the intermediate and long-term adverse events almost all occur between 30 and 45 days. So if you wait 60 days, you cushion it a little and overshoot a little. And if you don't see severe events during that time, it's extremely unlikely that you're going to see severe adverse events.
And just to clear up one point, my understanding is that these vaccines do not modify your DNA or RNA. So I think that's just an important point to clarify. If I'm getting anything wrong here, of course, correct me. But
No.
But just to make that clear.
No. First of all, DNA is inherent in your own nucleus cell. Sticking in anything foreign will ultimately get cleared. Messenger RNA is the messenger. It comes from the DNA to the RNA to the protein. Sticking an mRNA into you has absolutely nothing to do with your own DNA or your own RNA. It's going to perform its function, and then it's going to be degraded, and it's gone. It doesn't have any imprint on any genetic material of your own.
Good. Well, I'm glad we can clear that up. A number of people have also written in and asked, how do we get a clear answer on how long this immunity lasts? Right? If you're talking about, the effect going in and then potentially wearing off, does it look like this is the type of thing where we're going to need to get boosters regularly after the initial 2 doses, or do we think that this will be effective for a long time?
Well, Mark, one of the things that we have to be honest about in science is that when you don't know the answer, you say you don't know. And I don't know how long the immunity is going to last with the vaccine. When you look at immunity to COVID, to coronaviruses in the past, they're not lifelong immunity, unlike measles, when you get infected with measles or when you get vaccinated with measles. So we would hope it's at least a few years. And if it does ultimately require a booster, fine. I think the first step, given the situation that we're in right now, is to show that it actually works. So I'm hoping that the immunity will be durable enough to get us through a couple of cycles. And if it isn't, we'll cross that bridge when we come to it and boost people.
So one question that I'm seeing in the comments are, do the vaccines from Pfizer or Johnson & Johnson, for example, have any known collateral effects or symptoms Like this person mentions a fever in adults or anything else like that?
Yeah. Well, it is not uncommon, Mark, when you're dealing with vaccines, that there are immediate effects, and then there are the things that are more significant that might occur later. And that's the ones we spoke about that you can capture in that 60-day period. But with any vaccine, some more than others, when you get an injection of the vaccine, you induce a response. In some people, they don't feel anything. Others feel an ache in the arm. Some may feel an ache in the arm and kind of a little chilly feeling, almost like you have a flu-like syndrome. And in a minority of people, they'll get a fever. They'll actually get a fever for a day. Almost all of this goes away within 24 or at the most 48 hours. So we have to be honest when we tell people the adverse events, if you want to call them, are low grade. They're not serious. They're not prolonged. They're low grade.